Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 6, Microbial Genetics from our Microbiology A Systems Approach textbook, 7th edition. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
So first of all, remember that all living organisms have DNA as their heritable material. Prokaryotes, like bacteria for instance, typically have a single circularized chromosome. And within that chromosome, they have thousands of genes. Remember that each gene is a heritable unit that codes for a particular protein, or at least that's what a normal gene's function is to code for a particular protein. Here we're going to be looking at the genetics of microbes, looking at mainly the circular DNA of bacteria and looking at differences between prokaryotic and eukaryotic DNA. Again, remember that eukaryotes and prokaryotes possess chromosomal DNA. Eukaryotic DNA is linear and exists inside of the nucleus, a double membrane called the nuclear envelope. Inside, eukaryotes normally have several chromosomes, linear chromosomes. Conversely, bacteria typically have a single circularized chromosome that exists as a structure called the nucleoid. And that nucleoid is floating around in the cytoplasm. Notice how there is no nuclear envelope surrounding this DNA. The DNA is floating around directly in the cytoplasm. Again, eukaryotes have multiple linear chromosomes. Here's a chromosome, here's a chromosome. And bacteria have a single circularized chromosome. And of course, our chromosomes, whether it be eukaryotes or prokaryotes, consists of double-stranded DNA. When we refer to the genome, we're referring to all of the genetic material of an organism. Most of our genome exists in the form of chromosomes. For instance, humans typically have 46 chromosomes. However, remember, we also have what's known as extrachromosomal DNA. This is DNA that lies outside of the nucleus. This DNA falls inside various organelles, like our mitochondria. Do you remember when we discussed the mitochondria, we said that the mitochondria possess their own single circularized chromosome? Plants also contain chloroplasts in addition to mitochondria, and that chloroplast also contains its own single circularized chromosome. So these would be examples of extrachromosomal DNA. DNA that's not part of our nucleus, not part of our chromosomes, but is technically part of our genomes. In addition, several eukaryotes, but mainly prokaryotes, possess what are known as plasmids, tiny pieces of circularized DNA. These plasmids, we're going to go into these plasmids in more detail, but they can have important genes on them. They can have important genes with important functions for the organism. And these plasmids can be passed from organism to organism, giving different organisms these traits. We're going to talk about what plasmids are, but remember, plasmids are tiny pieces of extrachromosomal DNA that are typically found in prokaryotes, but can be found in eukaryotes as well. Now, the genomes of all cells consists of DNA. All living organisms have genomes of DNA. However, viruses, which are not alive, can contain genomes of DNA or RNA, but not both. And the study of genomics is the study of an organism's entire genome. Again, if we're looking at a eukaryotic cell, we have the nucleus, which is a double membrane comprising the nuclear envelope, and inside we have linear chromosomes. Humans would have 46 of these linear chromosomes typically. Outside of the nucleus, we're talking about non-chromosomal DNA, like the DNA found in our mitochondria, which is a single circularized chromosome, the DNA of plants and algae, which is again a single circularized chromosome, and various plasmids, which are tiny circularized pieces of DNA as well. In bacteria and in prokaryotes in general, you typically have a single circularized chromosome, which is called the nucleoid. And outside of that, you have this non-chromosomal DNA called plasmid DNA, small circularized DNAs with a handful of genes on them. And remember that viruses, which are not alive, can have their own genomes consisting of either DNA or RNA, but not both. Recall that a chromosome is defined as a distinct cellular structure composed of a neatly packaged DNA molecule. So think of it as a single double-stranded DNA molecule that's part of an organism's genome inside of the nucleus if it's a eukaryote, or as part of the nucleoid if it's a prokaryote. Eukaryotic chromosomes are located in the nucleus of the cell. They vary in number from a few to hundreds. Remember, humans have 46 chromosomes, but different organisms have different numbers of chromosomes. And eukaryotic chromosomes are linear in appearance. That means each chromosome has a beginning and an end. Remember, again, bacterial chromosomes. Usually, bacteria have a single circularized double-strand DNA chromosome, but that's not always the case. But for the purposes of this lecture, we're going to be discussing bacterial chromosomes as a single circularized chromosome. And remember the difference between genes and chromosomes. Genes are basic informational packets in which a chromosome is subdivided containing the necessary code for a particular cell function. In general, a gene is a segment of a chromosome that typically codes for a protein. For instance, humans may have 46 chromosomes, but those 46 chromosomes house 25,000 genes. You can think of a gene as just a segment of a chromosome that usually codes for proteins. That's not the entire picture, but we could simplify it to that definition for now. Also recall the terms genotype and phenotype. Genotype refers to the genes or various alleles that an organism possesses. Alleles are simply versions of a gene. So for instance, eye color would be a gene. Blue or brown eyes would be alleles of that gene. You can think of alleles as versions of a gene. And the alleles that you possess, that you inherited from your parents, make up your genotype. So for instance, my genotype might be brown eye color, and I inherited one of those alleles from each parent. So my genotype would be brown. However, your genotype might be brown-blue. You may have inherited a brown eye color allele from your mom and a blue eye color allele from your dad. So your genotype might be different from my genotype. However, the phenotype is the expression of the genotype. It's the trait that we see. For instance, if you see that I have brown eyes, that's my phenotype. Notice how phenotype is slightly different than genotype. My phenotype is brown eyes because I have brown eyes. However, my genotype could be brown because I inherited a brown allele from both parents, or brown blue because I inherited a brown eye color allele from one parent and a blue eye color allele from the other parent. However, prokaryotes like bacteria, they don't have parents because there is no sexual reproduction that occurs in bacteria, or prokaryotes for that matter, like archaea. And so a prokaryote would only have one gene or one allele for each phenotype. You don't have parents, so they don't have two sets of chromosomes. They don't have two alleles for every gene. Now, this leads to variation in genome size. E. coli, for instance, Escherichia coli, remember it has a single circularized chromosome. Well, that single chromosome houses approximately 4,000 genes. Humans, on the other hand, remember we have close to 25,000 genes residing on our 46 chromosomes. So different organisms have different numbers of genes, and genes typically code for proteins. Here you can see an actual image of DNA under a transmission electron microscope. Remember that DNA is made up of building blocks. These are monomers called nucleotides. DNA and RNA are made up of building blocks called nucleotides. Every nucleotide has three main components: a phosphate group, a deoxyribose sugar, and a nitrogenous base. The only difference with RNA is that there is ribose sugar instead of deoxyribose sugar. And in RNA, instead of the nitrogenous base thymine, we have the nitrogenous base uracil. Otherwise, the basic unit of DNA is the same for DNA and RNA. Again, in DNA, we have phosphate group, deoxyribose sugar, nitrogenous base. These three components make up the nucleotide of a DNA molecule. And if you've ever heard of the genetic code, you know how you may have heard that the genetic code is made up of A's, G's, T's, and C's. Well, that has to do with the nitrogenous bases of the DNA molecule. The nitrogenous base can exist as adenine, or A for short, thymine, or T for short, guanine, or G for short, or cytosine, C for short. These are the letters that make up the genetic code, and they are different versions of nitrogenous bases on these nucleotides. And when you string nucleotides together, this is how you obtain a strand of DNA. And you may remember from previous courses that each strand of DNA has a 5' end and a 3' end. And what's important to remember is that the two strands of DNA that make up the double helix of DNA, those strands are antiparallel. This means that, for instance, this blue strand of DNA has its 5' end here and its 3' end at the other end, but the complementary red strand would be going anti-parallel, would be going in the opposite direction, with its 5' end down here and its 3' end up here. So we say that double-stranded DNA consists of two complementary strands of DNA, and those two complementary strands are anti-parallel with one another. Remember that each strand of DNA has a sugar phosphate backbone. You see here highlighted in this peach color, there's the deoxyribose sugar followed by a phosphate group. Sugar, phosphate, sugar, phosphate, sugar, phosphate. This is known as the sugar phosphate backbone of DNA. And notice poking out the sides are the various nitrogenous bases, T's, G's, C's, and A's. And on the complementary strand of DNA, which is antiparallel, notice it also has a sugar, phosphate backbone and its own nitrogenous bases A, C, G, and T. The two strands are held together by what's called complementary base pairing. A's pair with T's with 2 hydrogen bonds. C's pair with G's with 3 hydrogen bonds, and so on and so forth. Remember when I said each strand of DNA, here you have 2 strands of DNA, each one has a 5' end and a 3' end. Well, recall that the 5' end of each strand of DNA has a phosphate group hanging off, while the 3' end of each strand of DNA has a hydroxyl group hanging off. Hydroxyl is an OH attached to the 3' carbon of this deoxyribose sugar. And there you have it, the double-strand DNA. I know that some of this may seem confusing, especially if you haven't had Biology 1406 or a Bio 1 course recently, because a lot of this that I just mentioned assumes that you've had some background in this. So if you haven't, just please make sure to read that part of the book so that you could become up to speed. Now, I told you that DNA or genes code for proteins, and that's typically what a gene does. It codes for a particular protein. So the process by which information is transmitted is that genes code for the proteins. Those genes are copied into what is known as mRNA or messenger RNA, which is essentially an RNA copy of that gene. And then ribosomes can interpret that mRNA message to produce a protein. This flow of information from gene to RNA to protein is called the central dogma of genetics. The first step to the central dogma is called transcription. This is where the DNA or the gene is copied to mRNA to synthesize a new strain of mRNA. Remember that mRNA stands for messenger RNA, and that messenger RNA is a single-stranded RNA copy of the gene. Next, during translation, that mRNA message is read by your ribosomes to produce a protein. That's what a ribosome does. You can think of a ribosome as a 3D printer that prints proteins based on the message found on the mRNA. And by the way, the process of making proteins from genes is called gene expression. The first step to gene expression is transcription, where a RNA polymerase copies the gene from DNA to a copy mRNA, messenger RNA. And the second part of gene expression is called translation, where ribosomes chomp down and they read the message of the mRNA from 5' to 3', synthesizing a protein based on that message. So again, what is the central dogma of genetics? The central dogma of genetics states that information flows from gene to mRNA to protein. Here you can see gene expression at work. We're starting with the circularized chromosome of a prokaryote, like a bacterium. A gene is then transcribed. A gene is copied into mRNA. Let's say this example here in the center, messenger RNA, which is then translated by the ribosomes into a protein. Remember that RNA polymerases are responsible for transcription, while ribosomes are responsible for translation. So again, we say that genes give us our traits, but that's only part of the story. It's actually the proteins which give us our traits. The genes merely code for those proteins. So for instance, if I have brown eyes, it's not because the gene is directly causing my eyes to be brown. What's happening is the brown eye color allele is being transcribed by RNA polymerase into messenger RNA, and then ribosomes read that messenger RNA to produce a protein. That protein produces pigment in my eye and makes my eye appear brown. So it's the protein that results from my brown eye color allele or gene that's actually making me have that trait, making my eyes brown. So you can think of it as proteins are what really give you your traits, but the only reason your cells know how to make those proteins is by having the genes and expressing those genes into proteins. So if that's true, then proteins ultimately determine your phenotype, how you look, what traits you have. And there's a whole new field of biology known as proteomics, which is the study of an organism's complete set of expressed proteins. We want to know what all of those proteins do in your body. And again, it's your DNA that is the blueprint that tells the cell how to make those important proteins. Again, to simplify and overview gene expression, remember that genes consist of double-stranded DNA. This is simply a segment of your chromosome. That gene is made up, again, of double-stranded DNA, but the template strand is the strand that is copied by RNA polymerase into a single strand of mRNA, messenger RNA, shown here in green. And it's then those ribosomes which can read through this message, this mRNA message, to produce the proteins. And do you recall that proteins are made up of building blocks called amino acids? There are 20 important amino acids that make up the proteins of your body. Those amino acids are strung together by the ribosome to form a chain of amino acids. And that is what a protein consists of. Now, I want you to note here that every 3 nucleotides on the mRNA transcript, see nucleotide 1, 2, and 3, 1, 2, 3, 1, 2, 3, every 3 nucleotides on the mRNA transcript code for a particular amino acid. And these blocks of 3 nucleotides has a name. They're called codons. And various codons are deciphered by your ribosomes to construct your proteins. In fact, let me show you this. This is known as the codon table. And notice that there are 64 3-letter words in the genetic language, 64 codons, if you will. So for instance, CCU, the codon CCU is translated by your ribosomes from mRNA to mean the amino acid proline, whereas UCA is deciphered by your ribosomes as the amino acid serine. Notice how you have these 3-letter words, these codons, and each codon has a meaning. Most codons code for amino acids, but there are a handful of very important codons. Let's start with this one here. This codon, which reads AUG, is a special codon. The first AUG that the ribosome encounters is called the start codon. It instructs the ribosome to start making a protein. And not only does it do that, but it tells the ribosome that the first amino acid should be the amino acid methionine. So yes, every protein should start with methionine because the start codon itself instructs the ribosome to put down the amino acid methionine. The ribosome will then read every codon between the start codon until it reaches a stop codon. The stop codons, there are 3 different ones, but any one of the 3 is sufficient to stop translation. The stop codons include UAA, UAG, or UGA. Any one of the 3 will tell the ribosome to stop making a protein. So isn't that neat? The entire genetic code can exist on a postage card. It's 64 codons, and those codons make up the genetic code. They make up the genetic language. They're responsible for all the proteins which make up all the diversity of all of life. Isn't that neat to think about? And the ribosome is the translator of the code. It knows how to read the genetic code and to build the correct proteins which are prescribed. Now for the first major difference I want you to note between gene expression in eukaryotes versus prokaryotes. Take a look here. Remember, this is a eukaryotic cell, and remember that the chromosomes are within the nucleus. This means that transcription occurs in the nucleus first. That's where the genes are transcribed to mRNA. And then, if you remember from an early biology class, Biology 1406 for us, at that point, that mRNA needs to be processed. Do you remember the 3 steps to mRNA processing? Splicing out of introns, adding a 5' cap, adding a 3' poly A tail. This is known as RNA processing, and it has to occur after the mRNA is formed. Then that processed mature mRNA is then allowed to leave the nucleus, meet up with the ribosomes, where translation can finally occur in the cytoplasm. Did you follow that? Transcription and RNA processing occur first in the nucleus, and then translation occurs later on in the cytoplasm. Now, I want to show you something. In a bacterial cell, the nucleoid, the genes are right there in the cytoplasm. So transcription and translation happen in the same place. They happen in the cytoplasm. There's no separation of the DNA from the ribosomes. They're both in the cytoplasm. And not only does transcription and translation happen in the same place, the cytoplasm, but it happens at the same time because RNA processing is not necessary in bacteria. So let me show you an image depicting what I'm talking about. On this slide, we have the protein assembly line of bacteria. This is what I'm talking about. Take a look here. Remember that the DNA is floating around as a nucleoid in the cytoplasm. The RNA polymerase enzyme copies a particular gene into a transcript. In orange, you see this is the mRNA transcript, it's called. And look what's happening. The ribosomes, which remember ribosomes consist of a large subunit as well as a small subunit. The ribosomes chomp down on the 5' end of the mRNA, and then they proceed to search for that start codon so that they can start making a protein. Notice how transcription and translation are occurring at the same time in the same place. Notice how transcription's not even done yet. RNA polymerase is still doing its job. RNA polymerase is still constructing a strand of mRNA. Before RNA polymerase is even done making the mRNA, the ribosomes are free to attach and search for the start codon to begin translation. Isn't that neat? So this should also tell you something else. Not only can transcription and translation occur at the same time in the same place, but RNA processing is not necessary in bacteria. This means there's no removal or splicing out of the introns. There is no 5' cap. There is no 3' poly A tail. The transcription and translation occur at the same time, same place, and it's very efficient, isn't it? In fact, it's so efficient that when the ribosome attaches and starts reading the mRNA, the next ribosome can attach, and then the next ribosome attaches, and then it pretty much becomes a Congo line of ribosomes. And so, like I said, before the RNA polymerase has even completed transcription, a Congo line of several ribosomes, this is called a polyribosomal complex, can attach to the mRNA, and they all search for that start codon and translate that message to protein here in purple. This is the protein in purple until they reach this stop codon, any one of the 3 stop codons, in which case they have produced protein. So protein synthesis in bacteria is very efficient. Transcription and translation occur at the same time in the same place, and RNA processing is not required because, for one reason, bacteria do not have what are called introns. They don't have this intron junk DNA, they used to call it, or filler DNA inside of the genes to splice out. Isn't that neat? So gene expression in bacteria is very, very quick and very, very efficient. Now for our second big difference between gene expression in bacteria versus gene expression in eukaryotes. And this is the presence of what are known as operons. Operons are coordinated sets of genes regulated as a single unit. Operons are only found in bacteria and archaea, so they are specific to prokaryotic cells. And how does it work? What exactly does it mean to have a coordinated set of genes? Let me explain what an operon is at the board, and we'll come back to go into some details about different types of common operon found in bacteria. Now, before I explain what an operon is, let me show you how genes work in you and me, in eukaryotes. When it comes to a eukaryote, we have a gene. And remember, a gene is a unit of heredity that codes typically for a protein. And in order to express this gene, remember, there is an enzyme called RNA polymerase. RNA polymerase attaches in front of the gene. It's called upstream of the gene at a site called the promoter. Yes, Gizmo. Gizmo, are you making an appearance on a live shot here? Come here, Gizmo. Yeah, Gizmo. You guys are finally seeing Gizmo in the flesh. Gizmo wants out. He wants to go find Wicket and play. Anyway, just we did not expect this little break time with Gizmo, but this is my buddy Gizmo, and he wants out. So say bye, Gizmo. Come on, guys. All right. Go play. Go find Wicket. So sorry. Again, in you and me, we have genes. Genes code for proteins. The way they do is first by a process known as transcription, where the RNA polymerase enzyme binds to the promoter. and then proceeds down the gene, transcribing the gene as an mRNA. The mRNA is a single strand of RNA that is a complementary copy of the template strand of the gene. And then, remember, the next step, we have translation, where the ribosomes will construct protein from that mRNA message. Okay, and remember, this is known as gene expression, the first step being transcription, and the second step being translation. And remember what I mentioned, this gene codes for a particular protein, and that gene is transcribed to mRNA, which is translated to protein. And that makes sense. You and I are eukaryotes. Each gene is transcribed as an mRNA, which is translated to protein. But now let me tell you the difference in prokaryotes and the fact that prokaryotes have a variation on this concept known as operons. And what is an operon? So let me explain that next. Now, operon are only found in prokaryotes, and you can find them in bacteria. And what an operon is a series of functionally related genes, one right after another. So on your chromosome, remember bacteria have a single large circular chromosome. They might have 3 genes in a row, for instance, gene 1, gene 2, and gene 3. These might be 3 different genes, but they code for functionally related proteins. What does that mean? That means the protein product from this gene and this gene and this gene might actually all synergize and do the same function. They might all help produce the same component for the cell, or they might all help break down the same nutrient in the cell. Okay, so for instance, the most famous and well-studied operon is known as the lac operon or lactose operon, and it's 3 genes that all serve to transport and break down lactose. And what's cool is these 3 genes, for instance, might have a single shared promoter in front. So you see, instead of each gene having its own promoter like you and me do, it may have a single promoter upstream of 3 genes. This means that when RNA polymerase binds to the DNA and proceeds to transcribe it actually transcribes gene 1, then it transcribes gene 2, and then it transcribes gene 3, and then it stops. This means that you end up with a super long mRNA. This mRNA is very long because the mRNA is, even though it's one long mRNA, it has information for 3 different genes, which means it possesses the information to synthesize 3 different proteins through translation. So during translation, a ribosome might attach here, a ribosome can attach here, and a ribosome can attach here. And this ribosome will translate this protein. This ribosome will translate a different protein, and the third ribosome will translate a third protein. So effectively, we have synthesized 3 different proteins at the same time. Right? Isn't that neat?
And remember, these three different proteins may all have the same job, like transporting lactose and then breaking down the lactose, you see? So they all function in a similar way. They all serve to do a similar function. So there are different operon that we're going to learn about, like the lactose operon, or the lac operon for short. There's the arginine operon, or the ARG operon for short. There are many different operon, and now you know how they work. Now again, I want you to know that we do not find operon in eukaryotes. So you and I, we don't have such setups. We don't have three different genes that could all be transcribed as one long mRNA, right? That's what an operon is, and it's only found in prokaryotes.
Welcome back. So now you know what operon are. Operon are sets or coordinated sets of genes all regulated together as a single unit. This means that a really long mRNA is constructed, and that mRNA contains more than one gene, and those genes are functionally related. So let's talk about how these operon are controlled in bacteria. Operons can either be what are called inducible or turned on, or repressible, turned off. So let's talk about inducible operon and why they're important, followed by repressible operon and how they could be important for the cell as well. Inducible operon contain genes that make proteins that help metabolize nutrients. So what is an inducible operon? An inducible operon is an operon that is normally not expressed. It is not transcribed and translated normally. However, the operon can be turned on. That's what induced means. So again, the operon is normally off, but it can be turned on or induced. And it is usually induced by a nutrient for which those genes encode. And it is normally turned on by the nutrient which those genes break down. So I'll give you an example. The most famous and well-studied example of an inducible operon is called the lactose operon, or lac operon for short. If there's no lactose around, the bacteria has no reason to make the enzymes to break down lactose. However, what if lactose is around? We want to break down that lactose for sugar, right? Well, if lactose is around, the lactose can serve as an inducer, turning on the genes that break down lactose. Isn't that interesting? Again, imagine if you're E. coli and there's no lactose present. You don't want to spend the resources to express the lactose operon and spend all those resources to transcribe and translate the 3 lac operon genes to make the proteins that help break down lactose. You don't want to make those proteins if there's no lactose around. It's expensive to make proteins. It costs resources, amino acids, ATP. It costs the cell resources to make enzymes. Why make the enzyme that breaks down lactose if there's no lactose in the environment? You see what I'm saying? So typically, inducible operon contain genes that make proteins that help metabolize nutrients. Nutrients like lactose. These operon are normally off. So if there is no nutrient around, then the operon is off. But if the nutrient is around, like lactose is around, then that turns those genes on so we can digest that nutrient. Isn't that interesting? So again, enzymes needed to metabolize a nutrient such as lactose are only produced when that nutrient is present in the environment. So let me actually show you how this inducible operon works. Let me show you how the lac operon works, and then we'll talk about the repressible operon. The lac operon consists of 3 functionally related genes, in this case, gene 1, gene 2, and gene 3. All 3 of these genes help to metabolize lactose. In fact, one of these genes is the gene called beta-galactosidase, which results in production of the protein beta-galactosidase, and this is the protein enzyme that breaks down lactose. So the lac operon consists of 3 genes, all that help to break down lactose. The lac operon has what's called the promoter. Remember, the promoter is this stretch of DNA upstream of the genes on which RNA polymerase, the enzyme that reads DNA to synthesize mRNA, can bind. RNA polymerase binds to the promoter so that it can start transcription. However, there's a stretch of DNA between the promoter and the genes, and this stretch of DNA is called an operator. The operator can accommodate this guy. Look at this thing right here. This is known as a repressor protein. It's drawn kind of like a clothespin in this image, and I like that because imagine this repressor protein is a clothespin. If I were to attach a repressor protein to the operator, and just like this clothespin looks here on this line, I want to ask you this. If the RNA polymerase attaches to the promoter and then it proceeds to go this way, wouldn't it run into that repressor? And wouldn't that repressor get in its way and prevent it from traveling down the 3 structural genes and transcribing those as an mRNA unit? And that's exactly right. Look, the repressor protein binds to the operator, preventing RNA polymerase from proceeding down the operon. So this prevents transcription from occurring. This means that the operon is effectively off. And remember, these inducible genes are normally off. So how do we turn this operon on, which means allow RNA polymerase to do transcription? Let's talk about that. Look at step 2. If lactose is added to the cell's environment, it triggers events that turn the operon on. You see these 2 purple dots right here? These 2 purple dots represent the disaccharide lactose. And it turns out that the lactose itself, the nutrient itself, serves as the inducer for this operon. How can that be? Take a look. The lactose disaccharide, it directly binds to that repressor protein. And when it does, watch, look what happens. The repressor becomes inactive, which means upon lactose binding, this changes the conformation, changes the shape, if you will, of the repressor, allowing the repressor to leave the operator. Now, let me ask you this. If the repressor leaves because it binds lactose, wouldn't that allow the RNA polymerase to continue down the structural genes and transcribe gene 1, gene 2, and gene 3 as one long mRNA molecule? Well, it will. So now you have effectively turned on the lac operon by adding lactose, the inducer. The lactose inactivated the repressor, allowing RNA polymerase to transcribe the 3 genes. And remember, because this is an operon, all 3 of those genes are expressed as one single very long mRNA, which is then recognized by ribosomes. The ribosomes chomp down in front of all 3 of the genes in this long mRNA to form how many different proteins? Can you beat Wicket? How many different proteins are made from this single long mRNA? How many different proteins can be made? That's right, Wicket. As always, very smart cat. 3 different proteins. Remember, 3 different genes are expressed, or transcribed as one long mRNA, ribosomes will construct 3 different proteins from that long mRNA. And all 3 of these proteins, including beta-galactosidase, are important to not only transport the lactose, but to digest the lactose into monosaccharides, glucose and galactose. Isn't that neat? So now the E. coli or the bacterium, whichever bacterium it is free to digest that nutrient. And whenever lactose becomes deficient in the environment, let's say the lactose levels drop off, then the lactose will detach itself from the repressor. The repressor will again activate, bind down on the operator, turning off gene expression of this operon. Isn't that neat? So again, the lactose operon is induced by lactose, and it can be turned on with the addition of lactose. Remember, inducible operon are normally off if that nutrient is not present, but can be turned on in the presence of that nutrient. And why is that? So that we can conserve resources when we don't have that nutrient around. Now, before we discuss repressible operon, I realized it's time for a break time with our favorite cats, Gizmo and Wicket. Let's see what these guys are up to, and we'll be right back with repressible operon. Whereas inducible operon are normally off but can be turned on, repressible operon are normally on but can be turned off. That's what repressed means. It means to turn off. So their normal state is on. And usually repressible operon contain genes coding for the enzymes that synthesize a particular amino acid. Remember that there are 20 amino acids, and all 20 are necessary for building proteins. So bacterium, they need to make these amino acids all the time. However, if a bacterium comes across a windfall of a particular amino acid, then it may not need to make any more at the time, and it can shut off the production of the enzymes that produce that amino acid. So for instance, what if I have E. coli in a broth, and that E. coli is making a particular amino acid? What if I provide that amino acid, like I inject that amino acid into its environment, and now it has plenty of that amino acid? Well, in this case, the presence of that surplus of that amino acid can repress the operon for that enzyme to make that amino acid, because why waste the resources to make the enzymes that synthesize that amino acid if there's plenty of that amino acid in the environment? You see what I'm saying? If there's not much of that amino acid in the environment, you need to synthesize your own by expressing these enzymes. However, if you have plenty of that amino acid in the environment, you could save resources by repressing the operon for these enzymes. Here's an example of a repressible operon. This is known as the ARG operon or Arginine operon. So remember, as with all operon, you have numerous genes that are functionally related. In this case, this is the Arginine operon. So these genes are expressed to form proteins that function to synthesize the amino acid arginine, one of the 20 important amino acids for making proteins. So the way this works is the operon is normally on, which means RNA polymerase is free to bind to the promoter, proceed down and transcribe structural genes 1, 2, and 3. Remember, a very long mRNA molecule is transcribed, which contains the information for all 3 genes. Ribosomes attach to translate all 3 genes. And how many different proteins are made? Again, can you beat Wicket? That's right, as always, Wicket. 3 different proteins are synthesized. Check it out. Protein 1, protein 2, and protein 3 are all created from that mRNA. And remember, the function of these proteins is to synthesize arginine. Arginine looks like these little stars, and these arginine amino acids are free to be used in metabolism. They're free to be used to make more proteins, okay? So notice how the operon is on. And the reason the operon is on is because the arginine repressor, the arginine operon repressor is inactive. It's inactive. It does not bind to the operator normally. So how does this operon turn off? Let's discuss that. How is this operon repressed? Remember, this operon can be repressed if I were to spike in a bunch of arginine. Imagine if the microbe encounters a high concentration of arginine. So let's discuss what happens. If arginine accumulates, you have a high concentration of arginine. At some point, when you reach this high threshold concentration of arginine, arginine will bind to that inactive arginine repressor protein and activate it. Now, because of binding to arginine, the repressor is active. In this case, they call arginine a CoRepressor because it helped the repressor become activated. So the CoRepressor, arginine, binds to the inactive repressor protein and converts it to the active form. The active repressor serves as a clothespin. Remember, kind of like a clothespin on a dryer line. It binds to the operator, and look what it's done. It's gotten in the way of RNA polymerase. RNA polymerase might proceed, but it will hit the operator. It will hit the repressor, which is on the operator, and transcription will be off. This means that the arginine operon is now off. Transcription is blocked. Arginine synthesis is arrested. It makes sense. If there's plenty of arginine, we don't want to waste the resources to synthesize these arginine making genes. Okay, so again, this is an example of a repressible operon. So now let's discuss the concept of recombination in bacterial cells. Recombination is an event in which one bacterium donates DNA to another bacterium. Remember that bacterium, they don't reproduce with sexual reproduction. However, they can exchange DNA, and this is known as recombination. And the end result of recombination is a strain of bacterium that's different from both the donor as well as the recipient. Oftentimes, the DNA that's shared is a plasmid. Remember, a small extrachromosomal DNA that can move between cells, but it doesn't have to be a plasmid. Bacteria can also share genomic DNA as well. So the term recombinant refers to any organism that now contains and expresses genes that originated in another organism. We discussed this briefly before. Remember that animals reproduce by sexual reproduction. When the male and the female mate, there is an exchange of genetic information, and that offspring has genes from both parents. That passing on of genetic material from parents to offspring is called vertical gene transfer. But remember that bacteria can donate genetic information from one bacterium, the donor, to another bacterium, the recipient. And remember, this is not called vertical gene transfer. This is known as horizontal gene transfer. And we briefly mentioned this in a previous chapter. Remember when we talked about the sex pilus and how certain bacterium can form a sex pilus and then send some genetic information to the recipient cell through that hollow tube? That's an example of horizontal gene transfer. Any transfer of DNA that results in organisms acquiring new genes that did not come directly from parents. So again, bacterium, they share genes with horizontal gene transfer, whereas animals, we share our genes through vertical gene transfer. Now, the sex pilus and conjugation are not the only form of horizontal gene transfer. So we're going to talk about the 3 different forms of horizontal gene transfer, because that's not the only one. And again, oftentimes it's this small circular piece of DNA called the plasmid, which is shared from one bacterium to another. These plasmids, they contain at most just a few dozen genes. But those genes may confer some advantageous traits to that organism. What kind of advantageous traits? For instance, the F plasmid, or fertility plasmid as it's known, is a plasmid that encodes the genes necessary for constructing your own sex pilus, for constructing your own sex pilus for conjugation. You can also have what are known as resistance plasmids, or R plasmids. These confer antibiotic resistance to the bacterium that possess them. There are also virulent plasmids for toxins and such, where the plasmid may contain genes that encode toxins. These plasmids often carry useful traits. Now, remember I mentioned there are 3 types of horizontal gene transfer in bacteria, and we touched on conjugation. However, there are 2 more forms, transformation and transduction as well. All 3 are common forms of horizontal gene transfer in bacteria, so we're going to discuss each. The first type of horizontal gene transfer is called conjugation. Remember, this is done through the sex pilus, and through that pilus, either a plasmid or other genetic material, which can include a portion of the genome of the bacterium, can be sent from the donor to the recipient. Now, it's important to understand that this is a one-way exchange of DNA, not a two-way exchange. So, DNA is sent from the donor cell, the cell that constructed the sex pilus, and that DNA is sent to the recipient cell. The recipient cell does not send genetic information back to the donor. And conjugation can occur in both gram-positive and gram-negative bacterium. And one of the best understood conjugative plasmids is called the F factor in E. coli. Remember, the F factor is also known as the fertility factor. It's a plasmid that contains the genes necessary to construct the sex pilus. So here we can see conjugation using the F factor. Take a look here. There are 2 forms of conjugation with this F factor. There is what's called F factor transfer and HFR transfer. Let's go through each. For transfer of the F factor, here is the scenario. Here is the donor bacterium on the top, And here's the recipient at the bottom. The squiggly blue and purple lines here, these represent the genome of the bacterium. Remember that the bacterium have a single circularized DNA in the form of nucleoid. Here is the nucleoid of the donor bacterium. Here is the nucleoid of the recipient. And in addition, you see this small circular DNA. This represents the F factor, or the fertility factor, a small circular plasmid of DNA. And what's interesting about this F plasmid, again, is that it contains the genes necessary to make this bridge, to make the sex pilus. So the donor cell is called the F plus cell because it contains the F plasmid. This donor cell constructs a sex pilus, which docks onto a neighboring cell. And by the way, this neighboring cell does not need to be the same species. It could be a different species of cell. Now, once the bridge is made with the pilus, the plasmid is copied, and a copy of the plasmid is sent to the recipient cell. This is known as rolling circle replication. This is how the plasmid is copied, and a copy of that F factor is sent to the recipient cell. And remember, in conjugation, genetic information only goes from the donor cell to the recipient cell. And in this case, a copy of the F factor plasmid is given to the recipient cell. And look, at the end of this process, both the donor and the recipient have a copy of the F factor plasmid, and so both cells are now considered F plus. And what's really neat, if it's the F factor that is shared, this bacterium is now capable of making its own sex pilus, and it's now capable of conjugating with other bacterium of either the same or different species. And this bacterium can now share that F factor with other bacterium. Isn't that neat? So through F factor conjugation, the donor cell shares the F factor with the recipient cell, resulting in 2 F positive cells. All right, how is this different from HFR transfer? First of all, what is an HFR cell? HFR stands for high frequency of recombination cell. So a cell that is called an HFR cell is a cell where something special has happened. Look at this. I want to show you something. Sometimes the F plasmid, the fertility plasmid, can actually insert itself into the host's circular genome. Imagine if I were to cut and paste this circular plasmid into the circular chromosome of the host cell. Now look at this. That's what an HFR cell is. Here in blue you can see the genome, the circular chromosome of the bacterial cell. And notice what's happened here. The F factor has integrated itself or recombined itself into the genome. It's like cut and paste itself into the genome of the cell. Now, this is known as an HFR cell. And because it possesses the genes necessary for building the pilus, it forms the sex pilus. The sex pilus conjugates with a adjacent cell. Notice how this adjacent cell is an F minus cell. And look what happens through, again, rolling circle replication. The DNA is transferred from the donor to the recipient. And notice this. In this case, it's not just a plasmid which is being shared with the recipient, but the F factor as well as genomic DNA, a partial copy of the donor chromosome. This this cell down here is not just getting the information for making the sex pilus, but it is also obtaining genetic information, genes from the donor as well, which is really neat. Then, when conjugation breaks, the bridge breaks, the donated genes are there in the recipient, and sometimes those genes can recombine into the recipient's genome. And now this bacterium is now called a recombinant. It not only has the genes, the fertility genes for making its own sex pilus, but if you look closely, it's also picked up some important genes from the donor as well. It's a true new strain. And again, as I mentioned before, the F plasmid isn't the only plasmid in town. There are R plasmids or resistance plasmids, which carry genes for resisting antibiotics or other drugs. There are virulence plasmids, which have genes for toxins on them as well. So they can share all kinds of different traits with one another. So now let's move on to the next type of horizontal gene transfer. This type is called transformation, and it involves capturing naked DNA from the solution, the environment. During transformation, a bacterial cell will accept small fragments of DNA from the surrounding environment. So imagine taking DNA out of the environment and then incorporating that DNA into your own. Some cells are capable of doing this. The cells that are capable of doing this are called competent cells. Not all cells are able to take DNA out of the environment that happens to be floating around out there and bring it inside. Only what are known as competent cells. These are cells with the correct machinery to bring double-stranded DNA inside. So let me show you how this might work. Here on the left, you have a bacterial cell which is very damaged and it's dying. This cell, notice how its circular chromosome has been fragmented. The bacterial chromosome is all broken up. This cell is dying. This cell explodes and lyses. Remember, lysis means to break open. This bacterial cell is effectively dead, and its innards have poured out. Okay, the double-strand DNA has poured out. Now, if this bacterium, this is the recipient healthy cell, if it's a competent bacterium, it's capable of taking double-strand DNA in from the environment. See, the double-strand DNA is being taken in by this bacterium. And if that double-strand DNA is incorporated or inserted into the genome of this cell, then a recombinant is formed. See, this portion in light blue, this portion of the recipient is from the host cell, from the donor cell, And so this is a recombinant. And this form of transformation, this form of horizontal gene transfer is called transformation. Notice how no sex pilus was required. A cell died and released double-stranded DNA, and the competent cell took up that double-strand DNA and incorporated it into its own DNA. That's a different way of horizontal gene transfer. Now, the final form of horizontal gene transfer is called transduction. And what's interesting about this form of gene transfer is that it requires viruses, specifically bacteriophage. Remember what bacteriophage are? They are viruses that specifically infect bacteria. And sometimes they could accidentally pick up donor DNA. They could pick up some DNA from the donor cell, and they could deliver that DNA to a recipient cell. So let me show you how this might work. This is a generalized form of transduction. We're not going to go into specialized transduction in this chapter, but generalized transduction works like this. Let me show you how this might work. First, a bacteriophage. Remember, bacteriophage look like this. This is a T4 phage. It has this shape like a lunar lander, like a moon lander. This bacteriophage attaches to the bacterial cell, and it injects its DNA, its double-strand DNA, into this bacterium. This bacterium is now infected with virus DNA. At this point, the viral DNA takes over the host cell and it produces more viral components. So here, the host cell is now producing more viral DNA. It's producing viral proteins. But notice what's happening here. This cell is becoming sick. This host cell is now infected and sick. And that means that its circularized chromosome might fragment. So here you have a piece of the host's circular chromosome which has broken off. It's a separated piece of host DNA. And the next step for the virus is really to package and produce new virion, right? Virion are individual viruses. And what should the virus package inside? Look at this red swiggle. This red stuff represents viral DNA. When new viruses are formed inside of this host cell, packaged inside should be viral DNA. But look at this guy on the right. Look at this guy. He's accidentally going to package some of that separated piece of host DNA. See? So viruses should package viral DNA inside, but this guy accidentally packages host DNA inside. Now, when it infects the next cell, watch this. When this cell breaks open, this cell lyses or breaks open, these phage are free to go infect the next donor cell, right? The next cell. These phage are free to go and infect the next cell. And notice, viruses should deliver viral DNA to bacterium. But notice this. Is this virus delivering viral DNA to this bacterium? Or is this virus delivering genomic DNA from the previous donor? It's delivering the wrong cargo. It's not infecting the cell with viral DNA. It's infecting this cell with DNA from the previous donor by accident. This DNA might incorporate itself into this new bacterium, and now a recombinant is formed. You see how this could lead to recombination? Because this virus inadvertently introduced genomic DNA from the previous donor and not true viral DNA, and that led to a recombination event. Isn't that neat? And for the last part of this chapter, let's talk briefly about mutations and how they might affect microorganisms. Mutations are any change to DNA, any change to the nucleotide sequence in the genome. So anytime a G gets mutated to a C or a T gets mutated to a G, anytime there's a change in the genetic code in the genome, this is known as a mutation. Mutation is a driving force of evolution. The normal gene, as it is normally found in the population, is called the wild type gene. However, a mutated gene, once a gene is altered, it's known as a mutant gene, and that forms a mutant strain of bacterium. The mutant strain is an organism that bears a mutation. Mutations can change the function of the proteins that result from those genes. What are some causes of mutations? Mutations can be caused spontaneously. These are known as spontaneous mutations. And this occurs by random errors in DNA replication. So, for instance, when a cell wants to divide, remember the first thing the cell needs to do is copy its chromosomes, right? Now, imagine during copying of the DNA, mistakes can occur. These aren't very common because DNA polymerase has proofreading ability. However, mistakes can occur. So, just spontaneously, there is a chance that genes obtain mutations. They incur mutations when the chromosomes are being copied before the cell divides. And this is known as a spontaneous mutation. There's nothing we can do about that. Mistakes are made sometimes. However, induced mutations, as the name suggests, result from exposure to either physical or chemical agents that damage the DNA. These are known as mutagens, and often they're also considered carcinogens. These are factors that cause mutations. They induce mutations, and these mutations compound on top of the spontaneous mutations. Here are a list of common chemicals that are mutagens. In several labs, we work with ethidium bromide, which is used to visualize DNA in agarose gels. However, physical factors such as radiation can also cause mutations as well. UV light, for instance, can cause mutations by causing cross-links or dimerization between adjacent pyrimidines. For example, thymine-thymine dimers or cytosine-cytosine dimers. While ionizing radiation, for example, gamma rays or X-rays, these can cause double-stranded breaks in DNA, leading to mutation as well. Now, what's neat is DNA damage can be repaired by a process known as photoactivation. Photoactivation requires this enzyme called DNA photolyase, an enzyme which uses visible light to repair DNA damage caused by UV. So you remember those thymine-thymine dimers we talked about, or those cytosine-cytosine dimers? Well, this enzyme, DNA photolyase, uses light in order to repair those damaged areas. Isn't that neat? So now let's talk about how microorganisms might be used to determine whether a chemical is a carcinogen or not, or whether it's a mutagen or not. The Ames test is a great test, a microbiology laboratory test that you can use to determine whether a chemical is carcinogenic. And it works based off of this principle of spontaneous mutations versus induced mutations. And this is the way the assay works. This assay typically uses the bacterium Salmonella typhimurium because of the back mutation potential to synthesize the amino acid histidine. What does that mean? Hold on. So what this means is they have found a strain of Salmonella that cannot make histidine because it has a mutation in the histidine gene. So again, we are starting with a strain of Salmonella, which is called His minus or histidine minus, because this strain of Salmonella cannot grow on histidine. However, a single point mutation can convert this bacterium back to histidine positive. So this is a great species or strain. This is a great strain to study for this assay. Here's how it works. We take our Salmonella, which is histidine minus. It has a mutation in the gene that codes for the enzyme that produces histidine, I should say. You place these His minus genes and you spread them on a plate. This is a minimal medium which lacks the amino acid histidine. Now, bacterium should not grow on this plate, remember, because they are not able to produce their own histidine due to that mutation, and this plate lacks histidine, so bacteria should not grow on this plate unless they incur a mutation. Now, some bacteria do end up growing on this plate, and those are known as the back mutations. They're the ones that fixed the histidine mutation and are now His positive. And notice, this is your control plate where no mutagen or carcinogen was added to the plate. So these are what's known as spontaneous back mutations. Remember, there's a certain level of spontaneous mutation that occurs, and that's unavoidable. So this represents your background level of spontaneous mutation. Now we want to do our test plate. The test plate is similar to the control plate in that it has a minimal medium which lacks histidine, but it also has the test chemical mixed in, the potential suspected carcinogen or potential carcinogen mixed into the agar as well. Again, no bacterium should grow. However, in this case, notice there are a huge number of back mutations. There are many histidine positive colonies that formed, much more than your control spontaneous mutation plate on the left. If this number is greater on the right than the left, this shows that these were caused by induced mutations. So this is truly a mutagen. This chemical that you're testing is a mutagen. If, however, this plate on the right exhibited the same number of back mutations as the plate on the left, that would have suggested that this is not a carcinogen, that there's no more colonies on the test plate than the spontaneous colonies on the control plate, so that would be labeled a non-mutagen or a non-carcinogen. I hope this made sense. The Ames test is really neat, so if you've ever wondered, how do scientists figure out if a chemical is a carcinogen or mutagen or not? Well, this is one very important way. And with that, that leads to the end of chapter 6. Thank you so much for joining us. Let's go ahead and move on to the next chapter. Please let me know in the comment box below if you have any comments or questions, and I'll be glad to answer. We'll see you guys for the next chapter.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.